So I'm gonna try to make this go as fast as possible because I'm trying to make this video short, but it may take a little longer to explain certain things and make sure you understand the background behind it. Uh, we've covered all this in class repeatedly. You guys have presented it and taught it, so you should have a good idea what the should be. DNA provides instruction for building which type of biological molecule. I hope everyone's written an mRNA, and you may have written here then to protein. But certainly RNA. And really, you could have written all that, but the honest answer and the complete answer is RNA. Because the reason RNA is the best answer is that DNA provides a gene, provides instruction to produce some type of RNA. It's that RNA could be a mRNA, in which case it goes and is the message that goes and becomes a protein. Or B, it could be a tRNA, which then goes out and transfers amino acids to the ribosome. Or C, rRNA, which then becomes the ribosome. And ribosomes, remember, are enzymes that catalyze. Remember what catalyze means. It speeds up the reaction so it's actually doable in real lifetime, in a real reasonable time. A ribosome, enzymes that catalyze the making of peptide bonds. In other words, to make a protein inside. So ribosomes, that's what ribosomes do. And then, of course, you have some others like small nuclear RNA, which are RNAs that exist in the nucleus. They help to cut out introns, splice together exons that we discussed early on in the protein synthesis lesson unit. Here's DNA. Where is DNA located in a eukaryotic cell? I hope everyone put in the nucleus. Why do they go as far as to make sure that you said eukaryotic? Because in prokaryotic cells, they're in the cytoplasm. Which cellular structures are the machines that build proteins? At this point, as I just discussed, it should be a ribosome. And if DNA can't leave the nucleus, how do you get the message of DNA out of the nucleus? You're going to have to turn it into mRNA. That's the message, as we said earlier. So the message is in mRNA. But remember, genes transcription occurs for both for mRNA, tRNA, rRNA, and snRNA. They're all transcribed from some kind of nuclear data that's in the DNA molecule. All right. This figure shows the flow of information from DNA to a molecule called RNA in the nucleus, and then the creation of proteins in the cytoplasm here, and then the creation. Then we now know that RNA is similar but not identical to DNA. It moves from the nucleus to the cytoplasm, from the nucleus to the cytoplasm, and this is where the protein's made: DNA to RNA to protein. RNA is a nucleic acid polymer composed of nucleotides like DNA. However, RNA has the sugar ribose and nitrogen-based uracil instead of DNA's deoxy and thymine. So remember, in DNA, you have T and the deoxyribose and the T letter T. In RNA, you have the ribose sugar and the nucleotide uracil. Fill in the blank: DNA to RNA to protein. And it would be nice to put mRNA because that's the message if you're filling out the message. But all three of these, 1, 2, 3, and honestly all 4 are required to produce the proper polypeptide or protein. At least 3 differences between DNA and RNA. That should be easy by now. DNA is double-stranded. DNA is big, RNA is smaller. DNA has deoxyribose. And 3, DNA has T, where RNA has uracil, U. All right, that's the answer there. What is the name of the process that uses DNA as a template to make mRNA? At this point, I hope everyone knows it's transcription. Look at how the DNA and RNA complement each other. Which DNA base does the U in RNA pair with? It would be an A, right? A pairs with U. And how is it different from the base pairing of DNA rules? In DNA, it's A bases with T, pairs with T. So in DNA, it's this. In RNA, it's that. That's how they base pair. That's how they hydrogen bond together. What is the name of the process that uses mRNA to make the protein? We call that translation. Now, all these answers are not only in the book and in class, but you see it right here. This is actually in the diagram. There's a gene, there's a DNA molecule, there's gene 1. Gene 1, you take this little piece of gene 1, that's what you see here, 3' N, 5' N. You transcribe it into mRNA, where there's an A, there's a U, where there's a C, there's a G, etc. Then you translate it in the cytoplasm, 3 codons at a time, to make amino acids. These answers to these questions were right here in the diagram. How many letters of the mRNA code? How many letters of mRNA code for an amino acid? I hope you know it's 3. At this point, I think everybody knows it's 3. It's a triplet code. And you can see it here. There's 3 here. There's 3 here. There's 3 matching with 3. 3 matching with 3. These 3 are matched with these 3, etc. This is a tRNA. So many people have consistently misunderstood, just not referencing, not getting if you don't do the work, you're not going to understand what's going on. tRNA is carrying the anticodon. It matches with the mRNA inside the ribosome. So tRNA brings amino acid. As you can see, there's an amino acid chain here. This diagram shows little spheres, little circles, darkened in circles. That's a growing amino acid chain, the growing protein. The tRNA brings in the protein. There's the next one coming in for a landing. Here's the third one. As this one drops off its amino acid and the peptide bond is formed inside the ribosome, the tRNA falls off, and the next one comes in to add to the chain. This whole process is diagrammed right here in this beautiful diagram, which, by the way, no one used, or I think one group actually used it and used little squares to block off the names. So this is a really great diagram that kind of outlines everything. So in the nucleus, the chromosomes are opened up by RNA polymerase. The transcriptions occurs. You have mRNA. You have RNA. Depending on which RNA, if you have mRNA, it comes out and is the message for the ribosome, where the tRNAs come in and drop off an amino acid based on the anticodon matching. What is the purpose of transcription? To make RNA. Where does this process occur? Nucleus. Transcribe the following template into mRNA. A goes to U, T goes to A, C to G, G goes to C, G to C, A to U, T to A to U, C to G. Done. What is the purpose of translation? to make a polypeptide. Sorry, I have to go clear the halls. Lunch is over, and some students don't know how to follow directions. Okay, so to make a polypeptide, translation is the reason for translation. Where does this process occur in the cytoplasm? If the protein is staying in the cell, and the rough endoplasmic reticulum if the protein's leaving the cell or going into a vesicle or some other internal membranous organelle. In any, of course, that's in eukaryotes. In any cell, only some of the genes are expressed, turned on. In other words, only some are transcribed. So the word, some people said they were confused about the word expressed and what it means. I think it's clear here that the word expressed means turned on. Means gene is expressed, which means that RNA is made. If RNA is made from the gene, then it is expressed. So if it's transcribed into RNA, it's expressed. There's 3 kinds of RNA. As you can see here, 4 really. There's more than that, but at least these 4. tRNA, right? There's a protein coding thing that's going to make mRNA. When they say protein coding, it means it's going to make mRNA. tRNA gene is going to make tRNA. The rRNA gene is going to make rRNA, which is the ribosomal RNA. So this is going to make the enzyme that makes proteins in the cytoplasm. This is going to bring the amino acids over to the ribosome, and this is going to match the codon with its anticodon. And then the snRNA gene makes snRNA. So those are the 4 kinds of mRNA that they describe. The 3 big ones that you need absolutely to know that will be on the test is mRNA, mRNA, tRNA, and rRNA. And their definitions are right here, and I've said it just in the last slide. States that only, Model 3 states that only some genes are expressed. Why do you think this is true? Why would a gene be expressed and why would it not be expressed? I think we've discussed this in detail in class with numerous examples, but every cell of your body has all the genes that every other cell does, so they're all copies of one another. And so all the DNA is the same. But if you're going to make an eye, then you need different genes on than if you're going to make a foot, and you need different genes on than if you're going to make a, a heart. Not even just that. Even in the heart, you don't always need gene 1 on or gene 2. Sometimes you turn on the genes you need. You turn on the genes you need. Now, here's some more students who can't follow directions. Let me go get the door. So you have to, even in the heart where you have genes that are turned on that are not in the eye, there are proteins you need when your heart needs to do one thing, and genes that need to be turned on when it has to do another. So different genes, even within the same tissue, are turned on and off at different times depending on the condition. So in other words, you're turning genes on that you need, and you're turning genes off that you don't need. That explains what we call differentiation. The ability of one single cell, as I discussed in class, when your father's sperm and your mother's egg met and produced you, a single cell in your mother's womb, in your mother's fallopian tube, became over time this specialized human being with different tissues, different organs, etc. That one cell, all these cells in this creature have all the DNA, all the same DNA as that single original cell. All right. So it states only as some genes are expressed. Why do you think it's I just explained that. You could rephrase it in any way you want, as long as it gets to the point. Cells turn on genes that they need to make the proteins they need or RNAs they need. If you have too many tRNAs, then you need to turn that gene off. If you don't have enough tRNA, you need to turn that gene on. How many types of RNA are there, and what do they do? We already listed them. They're listed right here. There's these three for sure. snRNA, small nuclear RNA, stays in the nucleus and cuts up mRNA, takes out the introns and puts the exons together. But in any case, these three are the big ones, so make sure that you write those, and their definitions printed, typed here, so I'm not going to rewrite it. So as you can see, Apogeo always has pretty much all the answers built in with it. It's all focused on the process and not the memorization of ideas. Here again is the codon chart we've discussed in class, and we've done practice problems in class. You have a U. If you have the first nucleotide is a U, and it's UU, then it's phenylalanine. If it's UCU, it's serine, etc. So the first nucleotide, the second nucleotide's here, the third nucleotide's along this line. So again, I'll walk you through one more. If I had AUC, if that was the codon, if I had an mRNA that had A, and then there was a U and then a C sticking out, then I would go to the codon chart and I would say, well, here's A, there's my first one, there's U, my second one, and C is my third one. Why'd I pick this row? Because that's where the A is. It's in the A row, the U column, and within this box here, right? Oh, I'm using the wrong color here. This box here, I know my answers in this box. How'd I know that? Because it's the A and the U were the first two. So the AUC would be isoleucine. You don't need to know the name. You just have to put ILE. All right. That's how you read it. Let's go ahead and see so we can do some practice. What do the letters U, C, A, and G in Model 1 represent? They are nucleotides. Nucleotides. They are, in this case, they are ribonucleic acids. So this is RNA. This has an oxygen here on this carbon has an OH group on it. Where in deoxy DNA there is none. Here, this is the difference between whether it's single ringed or double ringed. This is the difference right here. This is your nitrogen space where you're going to put either the U version of it, the C version of it, the A version of it, or the G version. All the rest of these pieces, the ribose sugar, the 5-carbon ribose sugar, again, 1, 2, 3, 4, and 5. The 5-carbon sugar, the phosphate, that's phosphorus with 4 oxygens on it, all connected, and they would form a sugar phosphate bond if you put them all together in a polymer called an RNA polymer, or a polymer of nucleic acids called RNA. Single-stranded, not double-stranded, et cetera, et cetera. What do the letters UG? So I just said that. What are the abbreviations for? They represent amino acids. So these are amino acids. And these are nucleotides. And in this case, phenylalanine, isoleucine, alanine, glycine are all different amino acids. To remind you what amino acid is, you have NH2, you have a central carbon, some hydrogen sticking off it. There's a different R group, and then there's a C double bond O, OH. Every amino acid looks pretty much the same. This R group here, though, is going to be very different depending on which of the amino acids you have. Phenylalanine is very big, has rings. Alanine's small, glycine's smaller. Some are polar, some are non-polar. Some are very similar, some are very different. The language of mRNA is often described as a triplet code, explaining the significance of the reference because the codons are in threes. Notice 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. All these codons are 3 nucleotides long, hence the triplet code, 3 nucleotide code. Sorry, I have to deal with people talking. So if your child did not go on the incentive trip and you hear me say something to somebody, it's one of them that you hear in the background that should be getting work done. Not sure who it is, but now that I turned around, they're quiet, so I guess I'll go back to work. If an mRNA molecule had 300 nucleotides in a coding region of strand, how many amino acids would be in the polypeptide that was synthesized? Show mathematical work to support your answer. Now, this is a tricky question. I saw a lot of kids answer this the easy way, and I agree with you. I think it was a really good that I saw it, and I gave you the points if you did it. 300 nucleotides. Then they said, well, there's a triplet code, so divide that by 3. I would get 100 amino acids. Well, realistically, I mean, on the surface, that looks absolutely right, and it makes sense. And again, I gave everybody credit for it. The only problem with that is don't forget the stop codon. The stop codon does not have an amino acid associated with it. So if you have 300 nucleotides, at the end would be a stop codon. You've divided by 3. 3 is 100. That's correct. But then 3 of those 100, that would be 100, except the last one is a stop codon, so it would have only 99 amino acids. Now, I gave you credit for 100 because I like your thinking, but don't forget the stop codon does not have an amino acid associated with it. How many different codons or triplet codes are there? Code for amino acids, the amino acid proline. Now, to answer that, you'd have to find the codon chart, which is on the previous page, but I have copied and pasted it here. Proline would be PR. They wrote the word proline, but they also put PRO in parentheses. They don't expect you to memorize that PRO is proline. So you go and look for pro on the list. There's 1, 2, 3, 4. 4 minimum. I don't see pro anywhere else. I'm sure someone will correct me if I made a mistake. I would say 4. I think that's all very easy material. Compare all the codons for proline. What are the similarities and differences? This is a key component. This is a key part of the problem. I understand some of you copied questions from each other, so you probably didn't get this idea. But if you take a look at proline, the difference between all of these is just the last codon, the last place in the codon. We call this last of the triplet code, this last nucleotide, this position, this third position, we call it the wobbly position. It wobbles. But often you can change, please pay attention to this because this goes into what we discussed in a later part of the unit. When you look at mutations, you'll notice that you can mutate this third position and you're always pretty much going to get the same amino acid. That goes for this wobbly position here for serine. The last position, it really virtually does not matter what you what you change that third position to, as long as the first two positions in this case are UC, then they're always going to be serine. But here, it does make a difference, as you can see. UUU is phenylalanine, but the other two are leucine. So you have to be careful, but certainly you can see, even when the one that only has two of them are the same, you see that third position is wobbly. That third position still can be more than one thing. Now remember, from the previous diagram and the previous illustration, tRNA has an anticodon. And it's that anticodon that they're talking about here that can be different. So each one of these represents a tRNA. You have a tRNA that has AAA. That one is going to carry a phenylalanine, a PHE. How do I know that? Because AAA, the complementary strand, the complement to AAA is UUU. So this is the anticodon. So the codon is UUU. Y'all think I'm playing? So anticodon. So on the mRNA, you'd expect to see UUU. And that's going to give you, when tRNA matches, you're going to get phenylalanine at the end. So again, you can reference the previous diagram that did a much better job than my silly little drawing. But you see that they match. And it's, in fact, that is the key, is that each one of these tRNAs carries a specific amino acid, but that amino acid can be carried by more than one tRNA. So the one with the AAA at the end carries a phenylalanine, but also the one that has AAG also carries phenylalanine. So there's two of them that carry phenylalanine, two different tRNAs, remembering that each tRNA has its own gene that codes for it. All right. So compare proline. What are the similarities and differences? It's the first two are the same. Let me reduce the size of this. First two nucleotides are the same. It's the last position that's different. Do you need me? Yeah, smart pass out, and then let me see it. Very good. Go. Considering the mistakes can occur during transcription and DNA replication, what advantage is there for an organism to have multiple mRNA sequences to code for the same amino acid? As we discussed in class, I'm saying here to you again, that protects against mutations disturbing function, right? So if you have a mutation at that third position, then you don't have to worry about it. Then you're still going to get serine. For instance, if a mutation happens in DNA and turns the A to a G, and instead of a U, you're going to get a C, it still has serine. So a change in the DNA won't always change the protein. That's really important, because DNA mistakes happen, and you don't want a mistake to stop life from continuing or cause life insult or what we call disease. So to protect against this, we have this built-in safety mechanism that has this redundancy. In other words, this last nucleotide can be exchanged out for any of the other, and mistakes can happen at every third nucleotide, and you'll be okay. Using the mRNA codon chart in Model 1, complete the following. If this is DNA, what does the mRNA look like? Well, let's go ahead and do it. If this DNA, then RNA is A, then this is U, and that's G. Now I can tell you right now, kids consistently put T in RNA. Even some of my AP kids were doing it, mainly I think because they weren't paying attention, they just wanted to turn something in. I can't imagine those kids not getting this, since they've seen it twice now, 3 times really. And so in any case, the point is that A, where you see here A, the other one's T, you see in mRNA there's going to be an A. Anywhere there's an A in DNA, there's going to be a U, and then G, and then A, and then A, and then C. I'm sorry, that's not true. Then C goes to G, then C, because that was G. A goes with U, T with A, then C, G with C, T with A, C with G, A with U, C with G, and T with A. So that would be our RNA. What would be the polypeptide sequence? Well, AUG is methionine. That's our start codon AUG. GAA, and I'm going to go ahead and just use the pointer here. GAA is glutamine, GLU. GCC is alanine. UAC is tyrosine, TYR. And CAG is GLN. And UGA is stop. So this is stop, nothing. There's no amino acid, as I mentioned earlier. Now again, people that copied, they got credit for this. That's great. On the test, you're going to have to be able to do this. According to the table in Model 1, what amino acid is at the beginning of every polypeptide? Well, I hope you know by now that it's methionine. It's always going to be methionine. Now, that doesn't mean it's always going to be there, because you can change the protein after it's been made, but initially it always starts with methionine. It's just one of the amino acids. According to table 1, or I just said it, methionine, MET. I'm just going to put MET. You don't have to spell it or pronounce it correctly, just MET. Which the codons in Model 1 are used in all species on Earth with very little variation. What my scientists conclude from this. So the fact that all life on Earth, with very small differences between the extremely different unrelated organisms, use the same codon chart. This is evidence of evolution. In other words, really what it is evidence of relatedness. That no matter how weird the organism looks, a jellyfish, a snail, a worm, they are related to us. We have similar mechanisms of producing the same processes. And that is a big deal. We have to explain that in science. Just want to mention again, that often people say to me that, they don't believe in evolution. You don't have to believe in evolution. It's fine that you don't believe in it, but just make sure that you can explain that evolution, some of the evidence for the argument of evolution is that there's relatedness between organisms, that even the most diverse organisms shared some form of DNA or DNA processing. So that suggests that there's a connection between all life on Earth, a deep, fundamental connection. All right, so again, now we're at translation. That was all transcription in the nucleus. Now the mRNA is outside the nucleus and we're in the cytoplasm. This shaded area represents the ribosome, as you can see, this large and the small. Within the side of the ribosome, the mRNA gets read. As mRNA is read, AUGs, which is always the start codon, they're always at the beginning. Even if it's in the middle of the mRNA, the coding doesn't start until you get to AUG. Then in comes the anticodon UAC. Notice the tRNA is a T that's upside down, is carrying methionine. So transfer RNAs bringing in methionine in. The next codon, and remember we're reading in threes, so this is the first three, then this is the next three. So now UUG, what you'd expect from UUG is AAC. And so one of these is AAC. Well, that would be AAC. This is the anticodon, and this is the codon. So we'd expect methionine, then leucine. If we go to the next one, the next codon is going to be GCC. So we would expect the anticodon to be CGG. So one of these is CGG. CGG. The anticodon here, CGG, means alanine's the next amino acid that goes here in the third spot. The next codon is AGC. AGC has the anticodon UCG. UCG carries serine. UCG carries serine. The next codon reads CAU. CAU would have the anticodon GUA. GUA is carrying histidine. UAA is the last codon, and UAA is a stop. So there is no amino acid here for UAA because there's a stop, as we discussed earlier. So you can read all these codons in the codon chart that we just looked at. I'm not going to put it in here again, just because it's just a waste of my time right now, and we're in a hurry. But you can look at the codon on the other chart on your paper, on your version of the paper. You can do it yourself. These codons all match up. If you look up AUG, you're going to see methionine, MET. You can say UUG. You're going to find the leucine is matched with UUG, etc. All right. So as they leave, the tRNAs go and get some more, and they call it uncharged. These are charged because they have an amino acid on it. The uncharged one goes and picks up another amino acid, again, that matches, in this case, UAC. UAC would go back and collect methionine, and only methionine. This tRNA only picks up methionine. This tRNA only picks up leucine. How do I know? The anticodons match, see? This one only carries leucine. It doesn't carry any other amino acid. So as they come in and they match, these are peptide bonds. You make this long chain of peptide bonds, and we call that a protein. All right. So when we make, what are the three stages of translation? The three stages of translation are called initiation, which means starts. The second step, that's one. The second one is elongation, which means it gets longer. And then the last, define each of the terms used in, and of course, the third is termination. So it ends, it stops. So this is the stop codon. Elongation is when the peptide bonds are being more paired up. And initiation is when the AUG matches with its anticodon. Defines each term and used in your answers in part A as they are used in everyday language. So initiation means start. Elongation means you're adding more and more amino acids to the chain, making it longer. And termination means you stopped it. All right, let's move on. According to Model 2, when mRNA leaves the nucleus, to which cellular organelle does it attach? I hope by now you know, without a question, that would be a ribosome. If you don't know that, I don't know what else to say. mRNA attaches the organelle at the sequence AUG. What is the significance of this sequence of nucleotides? It is the start codon. The process of initiation starts. Describe the movement of a ribosome as translation occurs. The ribosome moves one codon, 3 nucleotides. Remember, this is a triplet code. A codon means 3. At a time, matching tRNA to codon until there's a stop. Creating peptide bonds. Now, in between, a lot of you have already described this in your own words in front of the class. You did a great job. If you can't describe it in your own words, go back and review this. Review it in the video, on the reference videos that I've given you in class, in the ebook, in this diagram, and at the beginning of this POJO, whatever you need to do. The ribosome is a large complex of ribosomal RNA, and proteins. It consists of 2 subunits, a smaller subunit to the mRNA strand, and a larger subunit that holds the tRNA molecules in place. So the smaller one holds mRNA, so you have a small ribosome. They draw it like this usually, and the other one's a large one, and the large one holds the tRNA. And the large one holds holding the amino acid, and the small one is holding the mRNA. All right. While the covalent peptide bonds are formed in between amino acids, several ribosomes can attach to mRNA molecule simultaneously. This allows for many polypeptide chains to be made at the same time. So when we've been talking about one at a time because that's what makes sense. But if you have a long mRNA, you could have a ribosome that attaches now up here at the AUG, and there could be one that's halfway down the chain already. There's another one's a little bit further down, and the last one's a little bit further down. Each one of these can have a bunch of amino acids. It could be, and of course, every time as it moves it down 3 codon at a time, or sets of 3 at a time, the protein can get longer and longer until it finally falls off. So that's one way we can make protein really fast. Remember, this chain of amino acids held together by peptide bonds is called a protein. This is the beginning, and it gets longer and longer as it moves down the mRNA message. The tRNA molecules in the cell are short sequences of nucleotides, about 80 bases, that contain an anticodon, very specific, that carry a specific amino acid, which I've mentioned before repeatedly, I think, in this video, and in class, and on other videos. Find the tRNA model 2 that is carrying a histidine. What sequence of nucleotides makes the anticodon on this tRNA? Well, if you see there that histidine is being carried by GUA. That's the anticodon. So the answer is GUA. What codon mRNA would match this? Well, as I said earlier, it would be CAU. And if you look at CAU, one of these is CAU. Let's see, CAU. This is where histidine would go in the chain. Verify the codon you wrote in Part B codes for histidine by looking at the table in Model 1. As I said earlier, that's what I would have expected you to do, but they're actually asking you to do it, so let me pull up the chart. Now, if you look at at CAU, which is the we said here that the the codon for the anticodon GUA is CAU because they match. They would pair up that way. And we're saying that it would theoretically have to carry histidine. Well, if we look at CAU, what we see is it's histidine, HIS. So it is correct. It's verified. I have verified it. Check. What anticodon would be found on the tRNA molecule carrying glycine? So glycine, if you look here, there's no GLY here. How would I figure that out? How would I find the anticodon since it's not listed here, right? If it's not listed there, how would I find the anticodon? Well, they're asking the tRNA carrying glycine. There are a number of correct answers. You should know that because more than one tRNA carries glycine. We know that. Let's go ahead and take a look at it. GLY, no GLY here, no GLY here, no GLY here. There's GLU but no Y. There's GLN but no U, no Y. Then no GLY. Oh, there they are, GLY. So it could be GGU, GGC, GGA, or GGG. So the answer is going to be which tRNA carries glycine. It would be the anticodon CC, and then it could be CCA, CCU, CCC, or CCG. All of them can carry the amino acid glycine because that third wobbly position. But the anticodon would have to be CCA because the codon is GG. So the opposite, or the complementary, the complement to C is G. The T in tRNA is short for transfer. In the complete sentence, explain why this molecule is called transfer RNA, because it transfers an amino acid from the cytoplasm to the ribosome, so the amino acids can be linked. So it transfers our amino acids. That's the answer. I'm not going to write it down. I think you can rewind it and write it down yourself or put it in your own words, which would be better. Now, if you look at your ebook or you look at any online source, I quickly downloaded this as a good diagram rather than me having to write it in for you. Then what you can see is that there's three positions. The first, as the anticodon matches AUG, bringing methionine in. The ribosomal summary, the large and the small, come and match up. So there's your first amino acid. Then during elongation, the first one leaves, gets kicked out there, and another one comes in, another two. So there's the one that came in, and another one comes in right after it. So any given time during elongation, you get, any time during elongation, you're going to get two tRNAs in the ribosome at the time, until you get to the release factor at the end that attaches to one of the stop codons. Notice that the release factor has no amino acid attached to it. And in the end, you get this chain of amino acids that we call a protein. So you have initiation, elongation, and termination. So 2 is the answer, 2 at any given time. I am in the middle of recording. You're coming in. Sorry, excuse me. Stop. Why are you in here? Not in here. You're not. You go back wherever you're going. No. Where's your book bag? Why is it there? Because I put it there. Get out, please, and thank you. Okay. Goodbye. Okay. Thank you. All right, that's very good. I'm recording. It's still being recorded. I'm not editing this out. Have a good day. Have a good day. Close the door. Thank you. All right, moving on. During elongation, how many tRNAs molecules are held in the ribosome at a time? During elongation, how many tRNA molecules are held in the ribosome at a time? I hope you see this. It's 2. There's 2. You might say 3, but it's actually 2. One's leaving, the other 2 coming in. One's bonding, you need the other one that comes in and connects the 2 amino acids together. Well, one leaves. What will happen to to the unattached tRNA once it's delivered to the amino acid? Well, it gets kicked out. It's out it goes and gets another one, goes to get charged again, right? So it leaves and goes to get another amino acid. Describe two things that occur during termination as illustrated in Model 2. You get the bonding of the release factor, and then, so release factor binds, and then the ribosome falls off. The mRNA. There you have it. Explain how the term translation applies to the synthesis of proteins from DNA. So the way I look at it, when I look at translation, I think of changing a language, changing a phrase from one language to another. The language that you start with in the nucleus is in nucleotides. And they're in DNA nucleotides, all wound together in a double helix. We take that and we transcribe it. In other words, we rewrite it into RNA nucleotides. Still nucleotides, just a different form of them. So you have two sets. You have the coding strand, not coding strand, winding around each other. That's your DNA. Then RNA polymerase, the enzyme, comes in and reads it and makes RNA. But they're still nucleotides. When you get to the ribosome out here, the large and the small, and you take that that long RNA molecule, mRNA, and it reads it, and it puts together this long chain of proteins. Now you're talking amino acids. So you've changed languages. You've gone from nucleic acids, the nucleic acid language, to the protein language, or amino acid language. That's why I think of it. That's how it helps me think. Excuse me, where are you going? What? Did you write a smart pass? Oh, that's new to you? Okay. All right. So let's look at the extension questions. So when we're looking at how many permutations are possible, we have to consider what are the possible for each position. So there's 3 positions. Why are there 3 positions? Because it's a codon. Yes. Can we close that door, please? Since there's 3 positions, each position can have up to 4 possibilities. There's 4 possible answers. Four possible for each one. So you can have A, U, G, or C. Whenever you, what are the odds of getting an A in the first position? Well, 1 out of 4. Mm-hmm. What are the odds of getting A in the second position? 1 out of 4. What are the odds of getting AA? That'd be 0.25 times 0.25, and AAA would be 0.25 times 0.25 times 0.25. But so the difference, how many different permutations you have is you have 3 possible spots, 4 in each one, so it's 4 to the 3rd power, which is what?
That's 4 times 4 times 4.
Which is 64 codons. are possible. That's because each one can have one of four possible codons in a third one, and then there's 3 spots, so it's 64 codons. Show mathematically that two bases as a codon would not be sufficient to code for all 20 known amino acids. So 64. Did you write a smart pass?
Yeah.
Next time, could you show it to me?
All right.
64 codons. So 4 to the third power equals 64 codons. So now, if you only had 2 spots, if the codons were only, instead of a triplet code, it were a double code, then it would be 4, because you still have AGUC.
Yeah.
To the second power, which would only give you 16 options. It would give you only 16 codons. I'm not even going to call them codons because people get confused. 16 options. But how do you carry 20 amino acids with a specific series? You can't because you only have 16. You need at least 20. This gives you the 20 you need, but then it gives you extra. That gives you the added benefit, though, that if there's a mutation on that third position, you can have redundancy that protects you against mistakes. Mm-hmm. So win-win. You have enough codons for all 20 amino acids, and you can double them up. You have more than one tRNA carrying the same amino acid. So in case there's a mistake, you can protect against that mutation. A silent mutation is one that does not affect the protein structure. Write a code for the original DNA strand containing at least 12 bases, and then mutate the original DNA so that the final protein is unaffected. So we have A in the original DNA. We have A, T, G, C. A, T, G, C, A, T. We said 12, so that's 1, 2, position 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. The code is irrelevant for this discussion. What's important is that you know what the original was. So if we're going to make an mRNA to this, it's going to be U, A, C, G, U, A, C, G, U, A, U, A. Okay, well, how do I need to mutate this to get a silent mutation, right? So that my protein stays the same. So to do that, let's go ahead and put the protein, the amino acid sequence. And now UAC is the first, then GUA, then GUU, then UAA, AUA, sorry. UAC, I'm going to go ahead and practice this. UAC is tyrosine TYR. Out, no, out. Adam, out. Get out. Get out. No one comes in. Out. Please don't make me call security. I am in the middle of something. I'm not stopping the recording. Thank you. Close that door, someone. Thank you. UAC. So look at UAC. It's tyrosine TYR.
Leave the door open.
GUA is valent. VAL. CGU is arginine. And AUA is isoleucine. So that's what we have. And our job is to mutate the DNA, but keep these amino acids the same. Now, all throughout the video and throughout class, I've said the third position is wobbly. In most cases, if you change the third position, everything's going to be okay. So we can mutate the DNA that's associated with this one, this one, this one, or this one, and still probably be okay. We'd have to double check it, but let's go ahead and do it. What if I mutated this G to a C. If this G turned into a C, then this C would turn into a G. U, let's go ahead and follow it. U, A, G. UAG would be a stop, so that wouldn't work. That would give me a nonsense mutation. It would stop everything. So I can't have that. So that's not one of my options. That's one of those exceptions to the rule. This third position here is going to be a big difference. So now let's go ahead and, I could try to guess the second position. I could say, what if I mutate that? But I'm going to stick to the third position, GUA. And so CAT, I'd change the T to an A. What if I change this T to an A? So that would change this A to U. So GUA versus GUU. GUA is valine. GUU is
Also.
valine. They're all valine. So I can change this T to an A. So the new DNA code, to be clear, would be A, T, G, C, A, G, C, A, T, A, T. And I would still get valine in the end, even though the new mRNA would have GUU versus GUA. Because no, if you're not in here, you need to go. No one's allowed in here. Goodbye. We're working. We're going to work till 3. Thank you. In a prokaryotic cells, translation begins before transcription is finished. Give 2 reasons why this would not be possible. And why is it not possible that translation begin before transcription? Or, I'm sorry, yeah, that translation begins before transcription is finished. So here, all I really want you to write here, the only one I was thinking of, the only one I'm interested in, is that DNA is stuck in the nucleus. That's the big thing.